Good day everyone, I am Dela Cruz from Daizerie, and I'm here today to discuss the continuation of Chapter 6, which is all about the photosynthesis and the chloroplast. In our chapter outline, you will see the topics that we are going to tackle today. The first one is 6.6, the operations of photosystem 2 and the photosystem 1. 6.7, an overview of photosynthetic electron transport. 6.8, the photophosphorylation. 6.9, the carbohydrate synthesis in C3 plants. And the 6.10, the carbohydrate synthesis in C4 and CAM plants. Moving on to our first topic, which is the operations of photosystem 2 and photosystem 1. 6.6 terminologies. The first one is the photosystem 2. It is the first link in the chain of photosynthesis. It captures photons and uses the energy to extract electrons from water molecules. These electrons are used in several ways. So, si photosystem 2 is a process wherein na-absorb ng enzymes yung photons of light to energize the electrons. Then, yung electrons na yun yung magta-travel in series of cofactors and coenzymes to reduce the plastoquinone to plastoquinol. Then, next is the photosystem 1. Is a multi-subunit protein complex located in the thylakoid membranes of green plants and algae, which it initiates one of the first steps of solar energy conversions by light-driven electron transport. Kay photosystem 1 naman, it is a process wherein nare-receive niya yung electrons from PS2 or the photosystem 2, then eventually, ma-generate yon as the NADPH or nicotinamide adenine dinucleotide phosphate. In the next terminology na kasama sa 6.6 is the light harvesting complex, which is it is an array of protein and chlorophyll molecules embedded in the thylakoid membrane of plants and cyanobacteria, which transfer light energy to one chlorophyll, a molecule at the reaction center of a photosystem. Si light harvesting complex, ito yung nagta-trap ng light, then nagta-transfer nung light na yon or nung photons from light to photosystem para ma-convert as chemical energy. Next is the plastoquinone and si plastocyanine. Si plastoquinone transport electrons between photosystem 2 and the cytochrome B radical 6F complex, while si plastocyanine transports electrons between cytochrome B radical 6F complex and photosystem 1. So, si plastoquinone and plastocyanine, pareho silang mobile electrons na nagke-carry ng electrons to chloroplast para mabuo si electron transport chain. Next is si photolysis. Photolysis is the abiotic degradation of chemicals due to the radiant energy of light, either directly or due to reactions initiated by other light-absorbing compounds. Si photolysis siya nga yung pinaka-abiotic degradation ng chemicals due to the radiant energy of light. However, when it comes to photosynthesis, photolysis is the process of breaking water molecules using the energy provided by light, wherein yung molecules ni hydrogen, ni oxygen is mapaghihiwalay-hulay kasi kailangan ni photosystem yung electrons from the water para magkaroon ng electron transport chain. And the next is the oxygen evolving complex, which it is a supercatalyst to split water into molecular oxygen in the presence of light. So, si oxygen evolving complex, isa siyang cluster ng 4 molecule of manganese, 1 calcium, and 4 oxygen atoms, wherein tinutulungan nun yung process of photolysis na mas mapabilis and mas mai-transfer yung electrons sa photosystem 2. And the last terminology that we are going to use is the photoinhibition, where it is the process whereby light energy absorbed in the photosynthetic light processes exceeds energy demand of the dark processes, which leads to PS2 overreduction and subsequent inhibition of the photosynthetic capacity. Now that we are done talking about the terminologies that we are going to use in this discussion, let's start the discussion. The first topic is the photosystem 2 operations, wherein under this, the first operation is obtaining electrons by splitting water. The primary purpose of splitting water in the light-dependent reactions of photosynthesis is to release electrons for the electron transport chain. These electrons will then be utilized to create a proton gradient to synthesize ATP. The final electron acceptor of these electrons is NADP, which will form NADPH. So, sa process of obtaining electrons by splitting water, ito lang yung photolysis na nangyayari wherein paghihiwa-hiwalayan yung molecules na present kay water para mag-gain yung 4 na electrons na kailangan in the process of the photosystem 2. Next is the flow of electrons from photosystem 2 to plastoquinone. Figure 6.11, the functional organization of photosystem 2. A schematic model of the huge protein pigment complex which catalyzes the light-driven oxidation of water and reduction of plastoquinone. In the figure 6.11, makikita natin yung pinaka-process ng electron transport chain na nangyari under the photosystem 2. Absorption of this energy by P680 or the photosystem 2 excites an electron which is transferred to pyophytin, the primary electron acceptor of photosystem 2. Pyophytin is a chlorophyll molecule that lacks the magnesium 2 positive ion. The electron subsequently passes to a plastoquinone or the PQA and then through a non-heme ion 2 plus 2 plastoquinone B to form a negatively charged free radical plastoquinone radical B negative. And then, absorption of a second photon sends a second electron along the same pathway, converting the acceptor to plastoquinone radical B to negative. The two protons then enter from the stroma, generating the plastoquinone radical H2, which is released into the lipid bilayer and replaced by a new oxidized plastoquinone radical B molecule. So, kay photosystem 2 to plastoquinone, mag-start yung absorption ni P680 or ni photosystem 2 ng light or energy na nag-stimulate ng electrons, which is mata-transfer kay pyophytin. And then, dadaan yung electrons na yon sa plastoquinone A, then next sa ferrous ion, then to plastoquinone B para makapag-form ng negatively charged free radical na plastoquinone radical B. Then, pag nag-absorb ng pangalawang photon yung plastoquinone radical B, makapag-absorb din yon ng second electron para ma-convert si plastoquinone radical B to. Then, as we can see sa step 5, may another 2 protons na galing sa stroma para ma-generate yung plastoquinone na marirelease sa lipid bilayer. And magkakaroon ulit ng bagong plastoquinone radical B molecule. So, once na ma-generate na si plastoquinone radical H2, yun na yung tinatawag nating plastoquinol, which is a reduced form of plastoquinone radical B. And then, as we can see in the slide number 15, ito yung pinaka-conversion ng plastoquinone to plastoquinol, wherein, pag nag-absorb ng another 2 protons and another 2 electrons, yung plastoquinone, nare-reduce yung plastoquinone to plastoquinol. Kaya, ang magiging product niya is plastoquinol. And the next operation naman under the photosystem 2 is the flow of electron from water to photosystem 2. Sa flow of electrons from water to photosystem 2 naman, nag-o-occur yung pinaka-photolysis through the manganese-calcium cluster once na nakapag-absorb ng 4 photons of light yung photosystem 2. So, yung manganese-calcium cluster, di ba yun yung oxygen-evolving complex na binanggit ko kanina. Once kasi na nakapag-absorb na ng photons yung photosystem, pwede nang i-catalyze yung 4 electrons from the 2 molecules of water. Then, pag nakatalyze na yon, si manganese-calcium cluster, itatransfer nun yung 4 electrons through the tyrosine residue in the D1 protein as we can see in the figure, na yung tyrosine residue na yon, tinatawag siya as tier Z, papunta sa P680 dimer, which is the photosystem 2. And then, from there, mapapasa ulit yung electrons sa different parts of the photosystem 2 para ma-produce si plastoquinol. Then, next operation under the photosystem 2 is the flow of electrons from photosystem 2 to photosystem 1. As we can see in the figure 6.14, the electron transport between photosystem 2 and the photosystem 1, cytochrome B radical 6F operates in a manner very similar to that cytochrome BCl in the mitochondria and engages in a Q cycle that translocates 4 protons for every pair of electrons that moves through the complex. The plastoquinol 2 and the plastocyanin are mobile carriers that can transport electrons between distantly separated photosystem. So, moving into flow of electrons from photosystem 2 to photosystem 1, as I said earlier na yung plastoquinone is a mobile carrier. So, si plastoquinol from photosystem 2, nadi-diffuse siya through the lipid bilayer of the thylakoid membrane, then nagbabind siya specifically sa multi-protein complex na tinatawag na cytochrome B radical 6F, which napapasa doon yung pinaka-electrons na daladala ni plastoquinol. Then, the electrons from the cytochrome B6F or the B radical 6F will be passed to another mobile carriers called plastocyanin, which is nasa luminal side ng photosystem 1. So, si plastocyanin, dadalhin niya sa lumen yung electrons, and then from there, magta-transfer yung electrons papunta sa P700, which is yung reaction center pigment ni photosystem 1. So, moving on to the PS1 operation or the photosystem 1 operation, the production of NADPH. In the figure 6.15, the functional organization of photosystem 1. The path taken by electrons is indicated by the yellow arrow. Events begin with the absorption of light by an antenna pigment and transfer of the energy to a P700 chlorophyll at the photosystem 1 reaction center. So, the process of the production of NADPH, nag-start siya sa absorption of energy by the P700 causes the excitation of an electron and its transfer to a radical 0, which is the primary electron acceptor of the photosystem 1. The electron subsequently passes to a radical 1 and then to an iron sulfur center named F radical X. From F radical X, the electron is transferred through the two more iron sulfur centers called F radical A and F radical B, which are bound by a peripheral protein at the stromal side of the membrane. The electron is finally transferred to peridoxine, a small iron sulfur protein that is external to the photosystem 1 complex. So, when the two different peridoxine molecules have accepted an electron, they act together to reduce a molecule of NADP to NADPH. The electron deficient reaction center pigment, or the P700, is reduced by an electron donated by plastocyanin. So, moving on to the last part of the operations of photosystem 2 and the photosystem 1 is the photosystem 1 operations wherein dito mangyayari yung production ng NADPH. So, nagsisimula yung process nun once na nakapag-absorb ng light si P700 ng photosystem 1. So, from P700 mapupunta sa A radical 0, which is a monomeric chlorophyll molecule, which is the primary electron acceptor of the photosystem 1. Then, mapapasa yung electrons na yon to several cofactors starting from the quinone called phylloquinone or the A radical 1, and then two iron sulfur centers named F radical X and F radical A and the F radical B. Then, after makalagpas ng electrons from the iron sulfur centers, mapupunta naman yon sa peridoxine na small iron sulfur protein naman siya na nasa external part ng photosystem 1 complex. Then, para magkaroon ng reduction of NADP positive ion to form NADPH, magkakaroon ng catalyzing process using an enzyme called pyridoxine nicotinamide adenine dinucleotide phosphate reductase. So, pag nabawasan na ng molecule si NADP positive ion, magiging NADPH na siya, which is the reducted form of NADP positive ion. In the next slide, or the 23rd slide, basically, makikita natin yung pinaka-summarize ng process from the photosystem 2 to photosystem 1. An individual pyridoxine molecule can donate only one electron so that two pyridoxines act together in the reduction. Ang magiging product dito is 2 pyridoxine plus the NADPH. Then, we can write the overall reaction for photosystem 1 based on their absorption of 4 photons as was done for photosystem 2 as 4 electrons plus 2 hydrogen from stroma plus 2 NADP plus using the protons makakapag-produce ng dalawang NADPH. So, basically nga, summarize lang siya ng process of photosystem 2 and the photosystem 1. So, from photosystem 2, mapupunta yung electrons to mobile carrier called plastoquinol. From plastoquinol, matatransfer siya sa cytochrome B radical F, which mapapasa naman sa another mobile carrier called plastocyanin. And then si plastocyanin yung magdadala ng electrons papunta sa photosystem 1 para makapag-produce ng NADPH. Before we end the first topic, may question dito which is how are the two photosystem linked to one another? The electron transport chain links photosystem 2 to photosystem 1. And interruption in the transport chain would also halt NADPH production. So nakalink si photosystem 2 to photosystem 1 to each other because yung electrons from the photosystem 2 is ginagamit ni photosystem 1 para makapag-produce ng NADPH. And once na nagkaroon na interruption sa process ni photosystem 2, may interrupt din si process ni photosystem 1. So now, maybe merong mga nalilito kung bakit tinawag na photosystem 2 si photosystem 2 kahit na nauuna siya. Anong nangyari kasi doon? Unang na-discover ng scientist si process ng photosystem 1, kaya tinawag siyang photosystem 1. And then once na nagkaroon sila ng further discoveries about the processes na nangyayari sa plants, na-discover nila na ang pinaka-starting process talaga is the photosystem 2, which is yun yung nagpuproduce ng electrons ni photosystem 1. So, kahit na nalaman nila na nauunang process si photosystem 2, hindi na binago yung pangalan and binisa lang yung tawag sa kung ano yung unang na-discover and kung ano yung pangalawa. Kaya, si photosystem 2 siya kahit na nauuna siya na process ng photosystem. And then, moving on to our next topic, which is the 6.7, an overview of photosynthetic electron transport. In figure 6.16, we can see the summary of the light-dependent reactions, wherein sa unang figure, makikita natin yung 3-dimensional structures of the protein of the thylakoid membrane that carry out the light-dependent reactions of photosynthesis. One of the 4 major protein complexes, photosystem 2 and the cytochrome B radical 6F, are present in the membrane as dimers, whereas photosystem 1 and the ATP synthase are present as monomers. And then to the second image under the figure 6.16 pa rin, makikita naman yung summary of the flow of electrons from the water to NADPH, which is yun yung diniscuss ko kanina, that the process of the photosystem 2 to photosystem 1. Then, through the 3 transmembrane complexes. This figure shows the estimated number of protons translocated through the membrane as the result of the oxidation of 2 molecules of water, yielding 2 pairs of electrons. The ATP synthase of the thylakoid membrane is also shown. Approximately 4 protons are required for the synthesis of each molecule at ATP. Ito sa figure na to, makikita natin yung pinaka-summary ng process ng photosystem 2 and the photosystem 1. However, hindi na lang nag-i-stop dito yung process na yon, wherein may kasunod pang process which is madi-discuss later on. And then moving to the process or to the next topic, which is the 6.8, the photophosphorylation. So, photophosphorylation, the process of utilizing light energy from photosynthesis to convert ADP to ATP. Ito na nga yung makikita nating image kanina do sa may second image ng figure 6.16, wherein magkakaroon pa ng process para ma-convert si ADP to ATP using the NADPH na napuproduce ni photosystem. So, it is the process, or the photophosphorylation is the process of synthesizing energy-rich ATP molecules by transferring the phosphate group into ADP molecule in the presence of light. So, si photophosphorylation nga is a type of phosphorylation which includes the addition of phosphate in the presence of light or the synthesizing of ATP by cells. Nangyayari to sa chloroplast ng plants wherein kino-convert dito yung solar energy into ATP or the adenosine triphosphate. So, under the process of photophosphorylation, merong two types yun. The first one is the non-photophosphorylations. It involves both photosystem 1 and photosystem 2 in a linear electron flow that results in the production of ATP and NADPH. In this process, water is split by photosystem 2, releasing oxygen and hydrogen ions. So, sa non-cyclic photophosphorylation, yung electrons niya is nanggagaling sa photolysis or sa splitting of water molecules. Then, makakapag-produce din ng ATP using the hydrogen ions which eventually makakapag-form ng NADPH. The next one is the cyclic photophosphorylation. In this process, plant cells just accomplished the ADP to ATP for immediate energy for the cells. This process usually takes place in the thylakoid membrane and uses photosystem 1 and the chlorophyll P700. Cyclic photophosphorylation happens only in the photosystem 1 and only ATP is produced. So, sa cyclic photophosphorylation, ang involved lang is si photosystem 1, which means na para makapag-absorb ng electrons, walang photolysis na nangyayari and nakakakuha lang siya ng electrons from the light harvesting complex ni photosystem 1. And dito ang napoproduce lang is si ATP. Under the cyclic photophosphorylation, the figure 6.17 simplified SIM for cyclic photophosphorylation. Absorption of light by photosystem 1 excites an electron which is transferred to pyridoxine and to cytochrome B radical 6F plus to cyanine and back to P700. In the process, protons are translocated to cytochrome B radical 6F to form a gradient utilized for ATP synthesis. Another cyclic pathway for electron transport that involves movement of electrons from photosystem 1 through NADPH to cytochrome B radical 6F is not shown. So, ang pinaka-process niya is from absorption of light, magta-transfer siya to cytochrome and then to plastocyanine, then back to P700. Sa process na to, nata-translocate yung protons sa cytochrome to be utilized for ATP synthesis. Moving on to our next topic, which is chapter 6.9, the carbohydrate synthesis in C3 plants. So, ano nga ba si C3 plants? C3 plants go through the Calvin cycle, taking in carbon dioxide through the leaves' minuscule pores called stomata. An enzyme called rubisco helps the carbon dioxide combine with sugar. Eventually, the sugar and carbon dioxide form a molecule with 3 carbon atoms or C3 in the chloroplast. So, C3 plants, tinawag siyang C3 plants kasi once na nag-undergo siya ng Calvin cycle, may na-form na 3 carbon atoms. So, under the C3 plants, mayroong figure 6.18 which is the chromatogram showing the results of an experiment in which algal cells were incubated for 5 seconds in immersion in alcohol. One spot which corresponds to 3 phosphoglycerate or labeled PGA contains most of the radioactivity. Under the C3 plants, ginagawa nga yung process which is the Calvin cycle. So, Calvin cycle is a process that plants and algae use to turn carbon dioxide from the air into sugar. The food autotrophs need to grow. The reactions of photosynthesis that use the energy stored by the light-dependent reactions to form glucose and other carbohydrate molecules. So, si Calvin cycle is a process na ginagawa ng plants para ma-convert yung carbon dioxide into sugar, which is yun yung nagiging food nila. In this cycle, ang phase 1 is the carbon fixation, which is yung reduction ng high energy molecule na napuproduce ng ribulose triphosphate or the RUBP. Then next is the reduction, which is using the NADPH and ATP, nako-convert yung carbon dioxide into sugar, specifically glucose. And last is si regeneration, kung saan yung ibang na-produce na sugar and glucose is nare-recycle siya to regenerate the RUBP para magtuloy-tuloy lang yung Calvin cycle. And then, in the figure 6.19, converting carbon dioxide into carbohydrate, the first image, the reaction catalyzed by ribulose biphosphate carboxylase oxygenase or the Rubisco, in which carbon dioxide is fixed by linkage to RUBP. The product rapidly splits into 2 molecules of 3 phosphoglycerate or the PGA. And then, the second image naman is the abbreviated version of the Calvin cycle showing the fate of 6 molecules of carbon dioxide that are fixed by combination with 6 molecules of RUBP. The fixation of the carbon dioxide is indicated in step 1. In step 2, the I2PGA molecules are phosphorylated via ATP hydrolysis to form I2I 3-biophosphoglycerate BPG molecules, which are reduced in step 3 by electrons provided by NADPH to form 12 molecules of glyceraldehyde 3-phosphate or the GAP. Here, 2 of the gaps are drained away in step 4 to be used in the synthesis of sucrose in the cytosol, which can be considered the product of the light-independent reactions. The other 10 molecules are converted into 6 molecules of RUBP, which is makikita natin under the step 5, which can act as the acceptor for 6 more molecules of the carbon dioxide. And then last is the regeneration of 6 RUBPs requires the hydrolysis of 6 molecules of ATP. The NADPH and ATP used in the Calvin cycle represent the 2 high-energy products of the light-dependent reactions. And then, the next figure, which is, dito makikita natin yung pinaka-summarization ng process from the photosystem 2 hanggang sa Calvin cycle. So, photosystem 2 initiates photosynthesis by using light energy to create ATP and split water, releasing oxygen. The photosystem 1 uses light energy to produce NADPH. Both ATP and NADPH produced in these light-dependent reactions power the Calvin cycle, leading to the production of glucose. So, in this figure, we can see the summarization of the process from photosystem 2 to Calvin cycle, which nagsimula siya from absorption of light from photosystem 2 and photosystem 1, which nagproduce naman sila ng NADPH and ATP through the process of photophosphorylation. Then, yung NADPH and ATP na naproduce is yung gagamitin naman ng Calvin cycle para ma-process or ma-convert si carbon dioxide into sugar with the help of ribulose biphosphate carboxylase oxygenase or the Rubisco.
Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of 2 sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life, providing food and oxygen for the complex food webs on both land and in the oceans.
The next, is still under the chapter 6.9, is the redox control. So, the redox control is an oxidation reduction reaction, which is any chemical reaction in which the oxidation number of a molecule, atom, or ion changes by gaining or losing an electron. So, redox reactions are common and vital to some of the basic functions of life, including photosynthesis, respiration, combustion, and corrosion or rusting. In the figure 6.21, pinapakita yung redox control of the Calvin cycle. In the light, pyridoxine is reduced and a fraction of these electrons are transferred to the small protein thioredoxine, which reduces the disulfide groups of certain Calvin cycle enzymes, maintaining them in an active state. In the dark, electron flow to the thioredoxine ceases and the sulfhydryl group of the regulated enzymes become oxidized to the disulfide state and the enzymes are inactivated. Then next is the photorespiration, wherein photorespiration is a process which involves loss of fixed carbon as carbon dioxide in plants in the presence of light. Photorespiration is a process in plant metabolisms where the enzyme rubisco oxygenates RUBP or the ribulose biphosphate, wasting some of the energy produced by photosynthesis. So, si photorespiration is the process of consuming molecular oxygen and ATP which nagre-release naman ng carbon dioxide from organic substances in response to light. Photorespiration is harmful to seed tree plants because it reduces the plant's productivity, which is why it is also known as the wasteful process as the definition stated earlier. And in the figure 6.22, the reactions of photorespiration, rubisco can catalyze two different reactions with RUBP as a substrate. If the RUBP reacts with CO2, the reaction produces an oxygenase intermediate that breaks down into 3-PGN and 2-phosphoglycolate. The eventual outcome of these reactions is the release of the carbon dioxide, a molecule that the cell had previously expended energy to fix. In contrast, if the RUBP molecule reacts with carbon dioxide, the reaction produces a carboxylase intermediate that breaks down two molecules of PGA which continue through the Calvin cycle. Another one is the figure 6.23, the cellular basis of photorespiration, which is the electron micrograph of a portion of a leaf mesophyll cell of a tobacco plant showing a peroxisome identified by its crystalline core pressed against a pair of chloroplasts and close to a mitochondrion. The reactions of photorespiration that occur in each of these organelles are described in the next and in the text and shown superimposed on the organelles in which they occur. This series of reactions is referred to as the C2 cycle. The last steps of the cycle, the conversions of serine to glycerate in the peroxisome and then to 3-PGA in the chloroplast are not shown. Next is the peroxisome and the photorespiration. Peroxisome are small vesicles single membrane bound organelles found in the eukaryotic cells. They contain digestive enzymes for breaking down toxic materials in the cell and oxidative enzymes for metabolic activity. The most prominent role of peroxisome in photosynthetic plant tissues is their participation in the photorespiration, a process also known as the oxidative C2 cycle or the oxidative photosynthetic carbon cycle. So, si peroxisome, ang pinaka-prominent role nga niya is the participation in photorespiration because peroxisome contains all enzymes to carry out the photorespiration. And then, into our last topic, which is chapter 6.10, the carbohydrate synthesis in C4 and CAM plants. C4 plants, a plant that cycles carbon dioxide into 4 carbon sugar compounds to enter into the Calvin cycle. So, si C3 kanina, 3 carbon sugar, while si C4 plants naman ngayon is 4 carbon sugar compounds naman yung napo-form niya. And the C4 plants are very efficient in hot, dry climates and make a lot of energy. In figure 6.24, pinapakita yung structure and function in C4 plants. Electron micrograph of a transverse section through the leaf of a C4 plant showing the spatial relationship between the mesophyll and bundle sheath shells. Superimposed on the micrograph are the reactions of carbon dioxide fixation that occur in each type of cell. In step 1, carbon dioxide is joined to PEP by the enzyme PEP carboxylase in a mesophyll cell that is located close to the leaf exterior. The 4-carbon malate formed by the reaction is transported to the more centrally located bundle sheath shell where the carbon dioxide is released. The carbon dioxide becomes highly concentrated in the bundle sheath cell, favoring the fixation of carbon dioxide by rubisco to form 3-PGA which is shown in step 3, which can be circulated through the Calvin cycle. The pyruvate formed when carbon dioxide is released is shipped back to the mesophil cell, where it is converted to PEP. Although the process requires the hydrolysis of ATP, the high carbon dioxide or oxygen in the Baldel sheath cells minimizes the rate of photorespiration. Then next is the camplants. Camplants, a photosynthetic adaptation to periodic water supply, occurring in plants in arid regions or in tropical epiphytes. Camplants close their stomata during the day and take up carbon dioxide at night, when the air temperature is lower. Crasulian acid metabolism or camplants, also known as cam photosynthesis, is a carbon fixation pathway that evolved in some plants as an adaptation to arid condition. So the primary distinction between the C4 and camplants is how they minimize the water loss. C4 plants move carbon dioxide to molecules to reduce photorespiration, whereas camplants decide when to extract carbon dioxide from the environment. And into the last slide, the slide 59, we can see that the similarities and differences of the C4, C3, and camplants, as well as their structures. That's all for my report. Thank you for watching.